There are lots of claims that you can improve your child's destiny by selecting certain combinations of genes. I mean, this is not true, but people want to believe it. So you can see that there's a kind of, it's not a slippery slope, but there are gradations to this pre-implantation screening. And that's where the money is at the moment. So it's going ahead. But again, in the UK and in Europe, this would be completely illegal. You would not be able to do this.
Matthew Cobb, what do you think are some good examples of genetic engineering in everyday life?
Well, everyday life is perhaps putting it a bit strong, with one exception in particular, and that would be the use of insulin. So all of your viewers and listeners who use insulin, that insulin is made in genetically engineered microbes. And that was one of the promises that was made at the beginning of the genetic engineering revolution in 1975, and was very quickly realized, really quite amazingly. Within 2 or 3 years, it had been shown in the laboratory. And by 1980, Genentech, the little startup that was begun with a few $100s, it was set up the same week as Apple. In fact, they were both incorporated in the same place and in the same time. That went public on Wall Street and was at the time the biggest OPA that had ever been seen on Wall Street. It was soon eclipsed by Apple, but that gives you some idea of the kind of the domain that we're talking about. So that's the main use of everyday use of genetic engineering, and most people wouldn't even know. Before genetic engineering, insulin had to be obtained in a rather gruesome and long-winded way from the pancreases of slaughtered animals. And it wasn't actually, because it wasn't human insulin, it wasn't actually terribly good for you. And people would eventually have an allergic reaction because it's not identical to human insulin. And the advantage of genetic engineering was that they could actually make the actual protein, the real stuff, the real hormone, in a microbe. Then it's a bit more complicated than that, but by a series of steps, they could actually produce something that was exactly the same as that which we normally have in our bodies.
Mm-hmm. So we have these examples of how it is used regularly, genetic engineering, things like insulin, some foods, other processes we could talk about. So in your last book, you said that we're on a perilous quest with genetic engineering. What makes it perilous?
Well, I think the main perils have been that we've been very concerned from the very outset that there were possibilities that genetic engineering might inadvertently or advertently produce dangerous phenomena, so that you could either produce a microbe, for example, that was resistant to antibiotics, and this would be relatively trivial to do. And indeed, some of the early experiments very quickly, because people were using antibiotic resistance as a marker to see whether the experiment had worked, people then said, well, wait a minute, this could actually go horribly wrong. And this was in 1975 that people were concerned about this. There's also the concern at the time that we would be perhaps, if we were to introduce using, for various reasons, a virus that was thought to cause cancer at the time, if that was introduced into E. coli, which is a bacterium that lives in our guts, then we might run the risk of having an outbreak of cancer that would be transmissible. Now, these worries weren't realized. Today, I think there are rather different things we're concerned about, in particular. So again, they're well-meaning experiments that could go wrong. So one of the things that's particularly exciting and potentially life-changing is the idea of genetic engineering mosquitoes so that they would no longer be able to carry the malaria animal, the parasite that causes malaria, or other diseases that they can transmit, but that either they wouldn't be able to carry it or they'd go extinct. You could actually, by genetic engineering, make them unable to survive in the wild and actually eradicate them. And this sounds like a great idea. Why wouldn't you want to do that? You've got to remember that at the moment, something like 500,000 people die every year of malaria. Most of those are children under 5, and that's with all the insecticides, with all the bed nets that we use at the moment. So the idea of using this genetic engineering, it's called a gene drive, is actually very attractive. And I've spoken to many of the researchers who are involved, and they're extremely sensible and very, very careful and well-meaning people. However, we've got a basic problem in that we don't understand much about ecology anywhere, and in particular kind of tropical ecology. And we don't know what the consequences might be of, say, eradicating a particular species. People say, well, no animal survives solely on eating mosquitoes. That's true. But an awful lot of animals make a little bit of their living by eating mosquitoes, either as flies or as larvae, which live in the water. And we don't know what the consequences might be of upsetting the ecological balance. So that's something that's very concerning. And there's also been the two other things I'd mention. On the one hand, there has been, from the very beginning, there were attempts to, and they succeeded, to weaponize genetic engineering, in particular in the Soviet Union, to create novel pathogens for which there would be no defense. And so weaponizing this by military forces is something that is still very much a reality and a threat. And secondly, there is the prospect, which is not too on the agenda, but which nonetheless certainly excites some people and has actually been realized now, and that is of using genetic engineering to alter the human genome and to introduce changes that are transmissible. Having genetic gene therapy, that is, for example, being able to cure sickle cell disease, that is an absolutely fantastic therapeutic treatment, which is just becoming a reality. But that would not affect the next generation. So this is a genetic disease which is passed down the generations. And on the other hand, the possibility of introducing changes that will be permanent, that is something that was realized in 2017 by a Chinese researcher called Jiankui, who was very proud of what he'd done, but everybody else was aghast. And indeed, it all went rather wrong. And the 3 babies that were born by using this technique have had all sorts of odd mutations introduced into them. We don't know their state. I hope they're well, and I hope these didn't turn out to be of any danger. But this illustrates why it's perilous. These are very, very complicated procedures, and things that look simple on the chalkboard can turn out to be very complicated, either in ecological reality or in the reality of a growing embryo.
Yeah, you're opening a lot of threads that we'll have to discuss as we go.
Yeah, sorry about that.
It's great. It's great. In general, we're talking a lot about the social responsibility in the field. I think for those who don't know the history, I mean, what makes genetic engineering ethically different from all other fields?
Well, I think the main one is that, unlike every other field that I know of, I've been very insistent. This is the only example, and occasionally editors of articles have pushed back and said, "Can you prove this?" And I said, "Well, no, I can't prove a negative." But anyway, is that genetic engineering is the only science that has taken a look at its potential consequences and has pulled back from the brink and has said, we will not do this particular kind of experiment. That's happened 4 times since 1971, in fact, was the first time, even before it was a reality. So the very idea of doing a particular experiment was abandoned in 1971 when it was simply being sketched out because they were concerned at this, in fact, not real, but nonetheless at the time, frightening possibility that you might produce an epidemic of cancer by introducing these viruses into E. coli. So that was the first time. And then very famously, 3 years later, in 1974, when genetic engineering became a reality, and it became a relatively simple reality, then the scientists were again so concerned at the possibility. In particular, the thing that worried them was the idea of introducing a human gene into a bacterium like E. coli, with completely unknown consequences. Now, as it turned out, for various complicated reasons, and this was discovered within a couple of years, if you do that, nothing happens, because human genes are processed in a different way to the way that, they're structured in a different way to the way that bacterial genes are structured. And so the E. coli just doesn't know what to do with this message, which after a very brief bit of processing no longer makes any sense. But the scientists didn't know that at the time, and they were so concerned, they said, right, we need to have very strict rules, and we will therefore stop doing experiments until we can achieve that an agreement on how to do these experiments safely. So the interesting thing is that they were not saying, should we do them? They were saying, how can we do them safely? But nonetheless, this was eventually became known as a moratorium, and it went on for about 10 months until it was finally resolved in February 1975 at Asilomar in Pacific Grove near Monterey in California, where a meeting took place of around about 150 scientists, but also there were journalists there and some lawyers. And they came up, after a lot of fractious argument, they came up with a set of protocols which are still in use today. So they did pretty well, I think, for coming up with ways of doing dangerous experiments safely. And then the next two kinds of moratoria that came up was first in 2011, when a Dutch scientist was doing what is now a technique that is now sadly become well known, called gain of function research on H5N1. H5N1 is bird flu, which is very, very nasty, and were it to become transmissible to between humans, which at the moment it's not, you can get it by picking up a dead bird. So if you find a dead bird in the wild, or you're a farmer and you've got dead birds, do not touch them, right? Call in the relevant authorities, because they need to be dealt with in a very careful way, because there's a terrible bird flu pandemic going on at the moment. But H5N1 is not transmissible between humans. Were it to become so, this would be incredibly worrying, and it would make the COVID pandemic look like a tea party. It would be absolutely awful. So researchers were very concerned, and they wanted to know, was it possible that this particular virus could become transmissible? And if it were possible, then we would have a kind of early warning system. We would, because people are at the moment sequencing H5N1 from all over the world regularly to see what is happening. And if we knew what the dangerous mutations were, then we'd have a head start on the virus. That was the justification. So this isn't crazy. This is not tinfoil hat conspiracy theory stuff or anything like that. This is very sensible scientists. And they did the experiment using ferrets, which are the laboratory model for humans. If you can get one ferret to infect another, then you can be pretty damn certain a human will suffer the same effects. And it worked. And the scientist went to a conference and he said, I've done something, these are literally his words, I've done something really, really stupid. I've made H5N1 transmissible between mammals. And he was so alarmed at what he'd done, i.e. that it had worked, and other researchers were so alarmed that they had produced another moratorium. This went on for, in fact, they want it to be for a few weeks. It went on for nearly a year. And then there were lots of arguments about whether this was, funding arguments in the US in particular, which was funding all this research, whether this was a sensible thing to be doing, because clearly it is incredibly dangerous. And if there were to be an accident, if one of these modified viruses were to get out, then it could be very, very unpleasant. And the result was that, I don't think people aren't doing H5N1 research anymore. They've done that experiment. They know what it could mean. And they are, so we've got that early warning system. Whether it was a sensible experiment to do in the first place is another matter. The final example, when genetic engineering took this kind of choice, was, after Jiankui's experiment in 2017, people were so horrified by it that there was the immediate call for a moratorium in this kind of experimentation. Now, unlike all the other calls, which were either they were immediately effective and everybody who concerned accepted them, in this time it didn't happen. So, for example, Jennifer Doudna, one of the co-inventors of the CRISPR genetic engineering technique, she did not sign this appeal, whereas her co-inventor, Emmanuelle Charpentier, she did. So that gives you some idea that two colleagues who worked very closely on this, on the basic toolkit, could now not agree on what is the way forward. Doudna's argument is, well, the genie's out of the bottle. We'd be better off knowing what's going on rather than saying no. However, after a lot of rather slow discussion, the kind of biomedical community has currently agreed, but this isn't in any way legally binding or anything, that this kind of experimentation on human embryos should not go ahead because it is, at the very least, never mind the ethics, it's unreliable and dangerous, and the consequences will be cannot be foreseen.
Yeah, Matthew, it seems there's something unique about the culture of this field. To do a moratorium like this, or to do them over and over. You're a historian of the field. You've written the biography of Francis Crick. What do you think it is in the culture of the field that would make it this unique field?
Well, I think it's partly what happened in America, actually. So the scientists who were involved in this in the early 1970s were, in fact, not of the generation of Crick and the others who were all kind of in their 50s, 50s and beyond. They were much younger. So they were researchers who'd been involved, like Paul Berg, in the anti-Vietnam War demonstrations. They weren't radical in the sense of being, politically involved in any of the various organizations. So, for example, there was an organization at the time called Science for the People, which has kind of resurrected. It still exists. They weren't involved with that, but they were involved in teach-ins on their campuses against the war and so on. So they had and this was it was in the early 70s, but it was still the kind of the tail end of the hippie era, in which people were still concerned about social responsibility. We hadn't yet quite hit the 80s and the money, get rich quick world, which is, some of the same people were then involved in that as part of the intense dialectic of this period of history. But they were they were younger, they had a more radical outlook compared to many of the older researchers. And it's notable that Jim Watson, who was present at Asilomar at this big meeting, he initially signed the call for a moratorium, which was published in 1974. But by the time the meeting took place, about 8 months later, he changed his mind, and he said he'd been a jackass. Well, it's pretty true, but I mean, he'd been a jackass particularly to sign that letter, and he. Throughout the meeting, he kind of heckled everybody and said, look, this is all pointless. We should just go home. He was very much against the whole process. The reasons for that shift aren't quite clear, but it at least partially related to if you're going to do these experiments under these very stringent safety systems, somebody's got to pay for it. And he had just recently started running Cold Spring Harbor Laboratory on Long Island. And he was in charge of, what was going to become the big laboratory. And he didn't have any money to pay for this change. And it was unlikely that NIH, for example, was going to pay for it. So he was kind of more concerned, I think, with the nitty gritty of how are we going to do this. And he decided, having shifted his position quite substantially, that there weren't risks, or we couldn't measure them, and that with a bit of luck, everything would be okay. So there's a generational thing going on within the scientific community. The people driving it, people like Paul Berg and Hamilton, were much more, much younger and had a more radical outlook.
Mm-hmm. This sounds somewhat similar to what we're seeing today with artificial intelligence. I wonder, what parallels do you see or differences in how that's being handled?
Well, we can see an immediate similarity in that the AI community had a call for a moratorium on AI development, and then within about 2 weeks they'd all run after the money and said, forget about that, we can't stop it. And so I mean, this is part of the lesson of technology development, is that if you think about what the problems are after you've actually developed it and got it out in the wild, in whether it's in the laboratory or on everybody's apps, because they've all got AIs telling them things. It's too late, right? You've got to think about the problems before, or the potential problems, and then try and either work out ways of avoiding them or of preventing them occurring, or whatever, which is what the genetic engineering community did. Now, I think one of the reasons why the AI community has failed completely is because, well, NVIDIA for the moment, it's something like 16% of the US's GDP is NVIDIA. I mean, this is never mind about the personal wealth that some of these people are going to make. This is a huge, you might say, well, it's just the future of technology. You might say it's a bubble. That would be my advice. If I had any shares, I'd be selling. But there is immense. This is not just simply AI is not simply an academic issue anymore. It is now one about economic futures, about social futures, in a way that the genetic engineering discussions, at least those in the 1970s, were not. Maybe there's a bit of similarity with the issues over human genetic engineering, but that isn't a financial one at the moment. That is, there's no vast sums of money that's going to be made, because if this were ever to be realized, it would be incredibly expensive and also pretty grim for those involved. Well, at least for the women. For the men, it would be a few minutes in a cupboard and that would be it. For the woman, it would be involved going through IVF, which is not easy, no guarantee of success, pretty hard going, and then with an end result that you don't know. So I think there are very strong parallels, and there are certainly differences within the AI community, but I don't sense that there's a generational thing, partly because, we haven't had a 1960s since the 1960s. And the critics of the claims of AI, people like Gary Marcus, with whom I would have a great deal of sympathy, tend to be, actually part of the AI establishment. This is an argument that they've been having. In fact, they've been underneath it all. The argument goes back to the 1950s as to what artificial intelligence looks like, and what the best thing to do to progress the field. This argument has recurred over and over again in the history of artificial intelligence research, but now it has become one of incredible financial significance.
Yeah. It's interesting. You said we haven't had a 1960s since the 1960s, and yet I do see similarities between, say, Vietnam and the war in Iraq and Afghanistan.
Oh, yeah.
Yeah. And yet maybe Agent Orange was a factor in this, and because it was a biological thing with genetic engineering, as compared to we don't have something, and an AI element in Iraq, for example, that's as connected.
Yeah, I mean, that's an interesting point. I mean, Agent Orange was in fact extremely important in the development of what in the US anyway is the other main everyday application of genetic engineering, that's agriculture. So when Monsanto funded and helped to develop the first genetic engineering systems in plants in the mid-1980s, which was an awful lot of work, very complicated procedure, and involved people all over the world. So it wasn't just Monsanto. But Monsanto's aim was to get out of chemicals, because they made AstroTurf, they made Agent Orange, they made DDT. This was kind of the 1950s horror scheme for the ecosystem, was Monsanto. And their head, their CEO, said, we've got to get out of chemicals, because he literally said is not sustainable. The planet, neither the planet nor the general public will accept this. So the clever scheme they came up with is, rather than spraying insecticides everywhere, we will using genetic engineering, we will persuade the plant to produce its own very specific insecticide against caterpillars. So it's not a general insecticide, it's just if a caterpillar eats this plant, it will die. And that very, very clever, extremely well, again, very, very well-meaning aim was one of the most successful results in terms of agricultural change and shift in agricultural technology there's ever been. And it's also resulted in a massive reduction in the amount of pesticides, insecticides that are sprayed around the world. Now, evolution is smarter than we are, so there are now resistant forms of the various insects that are eating cotton and maize and all the rest of it. So this is, it's not a simple, you don't win once. It's a perpetual battle, and you need to be very smart about it. But I think that's an example of how Agent Orange, to use your reference, carried on in terms of AI. I guess at the moment, then nothing terrible we've seen has happened yet. Maybe if it were to happen, it would be so awful that we couldn't actually, we won't survive it. Not because they'll try and kill us, but because maybe they'll, you could imagine all sorts of terrible things happening with weapon systems or with the financial system. I mean, remember that, the financial system had to kind of put the brake on automatic trading because it was just going crazy and could lead to bubbles and crashes and so on. So I would be rather concerned about both those fields myself. Any involvement of AI in military matters or in the trading system would be a cause for concern.
Mm-hmm. And I think you said earlier that looking at safety sort of before putting things out there would be a good path. With the moratorium, it seemed early on it was about looking at safety, and now maybe discussion of ethics and things just slows it down. So as we're looking at things in the modern day, talking about AI, should we be trying to assess direct risk rather than broader theoretical things, or what seems to resonate with humans to make them slow down?
Well, I think one of the ways is when you can look at the immediate consequences. So theoretical risks are, they're exciting for theoreticians, but people tend not to. Most folk are more interested in things that they can actually see. So if you tell them that, your AI is going to, there's a big article just recently looking at the role of AI in recruitment. So if you give some of these large language models loads and loads of CVs and ask which one would you employ, then what do you know? It tends to overvalue the older male CV and undervalue the younger female CV. Now, this is not surprising because this is what HR people, not AIs, do all over the world. And the AI is simply reflecting the biases that already exist, but it's also amplifying them and automating them. So I think that kind of thing, we've got an example in the UK of them trying to be very clever. The government's trying to save money, and so they have restricted the benefits to people if they think. These are child benefits, which everybody gets. If you have a child, you get a certain amount of money from the state. And if the computer says, look, you left the country, but you never came back, therefore you've emigrated, therefore we will cut off your funding. And many of these cases have turned out to be cases of people who were going to catch a plane, but something happened. Their child was ill or whatever. They didn't get the plane. So the message on the computer says, they caught a plane, but they never came back. Whereas in fact, they've been living in the UK all the time. So those kind of injustices, clear injustices from a very simple, it may not even involve an AI. It may just involve a bit of rather clunky programming on Excel. Those kind of things are what alarm people, I think, or seeing things get out of control. There was also it was a few years now. I forgot what it was called now. There's a campaign against autonomous weapons, which was, and they made a fantastic video, which was a bit, this kind of Steve Jobs character showing you that he was making these, drones which would take out the bad guys because they were powered by AI. And the whole thing was absolutely terrifying and completely believable. We're getting there. So depends who are the bad guys, of course. Yeah. And whether you should take them out in the first place.
So to get a little more grounded in real risk, what have been some of the biggest close calls with dangerous gene editing?
I think the H5N1 was that was a close call. If they're frightened, we all should be.
And how does that compare to say, COVID-19?
Oh, I mean, I can't remember what the death rate of COVID-19 was, 5%. It'd be something like 50% with H5N1.
50%.
Yeah. I mean, that it would be absolutely catastrophic, which is why they did the experiment. I mean, I'm not a virologist. I'm not an epidemiologist. Okay. So I can have my opinions, but I also need to pay close attention to people who are very well-meaning. They're not crazy. They're very sensible and are very concerned. And their view in general is that this kind of experiment is warranted because of this early warning that it will enable us to have, which we've got now. So I know from seeing that the work that's being done that people are very closely looking at naturally occurring mutations in H5N1, because the thing about viruses we saw from COVID is they mutate incredibly quickly because they have very brief life cycles. So there's lots and lots of opportunities for new mutations to arise. And the vast majority of them are either negative or have no effect at all. But occasionally it just takes one to help the virus spread more rapidly or whatever, and we've got a real problem. And that's clearly what happened in the case of COVID-19 when it made the jump from the bats or the pangolins, the mammals that it was living in the wild, to humans, which is something that's happened over and over again. In fact, we can even see it in our genomes. We can see the remnants of responses to past pandemics because, we've lived in the forest and we've been close to animals, and viruses are very, very clever things. They will find a way of getting us. So I think that H5N1 experiment was probably the most alarming thing. Now, as I say, the researchers who work in this field and who are immensely sensible, they still think that this was a valid experiment to do with all the dangers that it involved, because it now enables us to know what is going to happen. On the other hand, it was also bigged up at the time as this is going to help us with the next pandemic. Well, none of that research helped us with COVID-19. It was a completely different line of research which enabled us to produce vaccines.
Yeah. Any other big scares that come to mind?
Well, I mean, we've had dangers, but it's not been from genetic engineering. So one of the reasons why people got so worried in the 1970s was that twice in the UK, we came as close as this to a smallpox outbreak. So smallpox is now, it's extinct, apart from those that we have in freezers, and which are occasionally rediscovered. So the smallpox was absolutely devastating disease, and it was eradicated through a program of vaccination run by the WHO, and is now completely extinct in the wild. We have some tubes of it, as I say, retained. I'm not quite sure why, but they say it will help us in the future. My personal view is they should be destroyed. But in the 1970s, twice, once in London and once in Birmingham in the UK, then there were lab accidents that led to people dying from smallpox. And in both cases, by very straightforward public health measures, which is also largely what restricted the spread of COVID-19 to a large extent, they were able to stop it from getting any further. But that could have been devastating. Now, then we were all vaccinated, so it wouldn't have been quite so worrying. It would only been the unvaccinated who would have died. But today, if such a thing were to happen, where very few people, it's only old people like me. I mean, I guess you probably, do you have a little mark on your shoulder from smallpox? Almost certainly not. Because why would you? It's been eradicated. So that is the thing that I think is most alarming. But that again, that wasn't an engineering issue. That was just simply an escape issue, and the procedures that were involved for keeping the virus, keeping it were very limited compared to what we would have today and the procedures that we have today for keeping control. I mean, there were also experiments that went wrong in the Soviet Union, engineered forms of anthrax, which escaped and killed hundreds of people in the Soviet Union. Whether they escaped, whether it was an explosion or whether it was a deliberate release, we still don't know. The Soviets said it was because of contaminated meat. But it wasn't. It was very clearly proved that there was a weapons facility downwind or upwind, whichever way, from the main center where the people died, and the wind blew the spores in their direction.
Mm-hmm. Yeah. So this is that gain of function research then. These sort of, we've got bad things, and then we're going to make them worse so that we can understand them. I mean, the logic there is to understand the sickness better. That's the idea. This is supposedly defense.
Yeah, well, yeah. I mean, you got to remember that all this goes back to this was very much advocated by the USA in the wake of 9/11. So in the wake of 9/11, there was the crazy guy who put anthrax spores in envelopes and sent them to people, and a number of people in the USA died. And the government, I mean, basically the US went mad. I mean, understandably so, but there was a huge wave of paranoia, in particular at the potential technical developments that terrorists might wish to employ. And with the various developments in genetic engineering, it started to become called synthetic biology. I mean, it's all the same thing, really. It's fiddling around with genes to alter microbial organisms. But developments in synthetic biology led the US government in particular to start pouring vast sums of money into gain-of-function research to see, well, quite how bad, if there were bad people, what could they do with various already existing pathogens. I mean, and sometimes these things were discovered by accident. So there were some Australian researchers who were trying to, using a disease called mousepox, which kills mice, right? So it's a bacterium that kills mice, and it's very closely related to smallpox. But they were interested in mousepox because they wanted to try and find a disease that could eradicate rodents, placental rodents, the kind you have in your city, in Australia, where there are a lot of marsupial animals that are being outcompeted and so on. So this was, these were conservation biologists, and they did a particular procedure, and they discovered that inadvertently they had now made the mouse immune to mousepox, and they were absolutely terrified because we're back to smallpox. In principle, the same thing would work with smallpox. You could make smallpox, rather than making the mouse immune, they made the mousepox able to escape the effects of the vaccine. Yeah. So that would mean that my patch on my arm would no longer work, because if you could do that to smallpox. So they were terrified because they realized they manipulated the mousepox in a particular way, and that this would mean that they could therefore, somebody using the same technique could develop similar ways of altering smallpox. So they worried about publishing it and so on and so forth. In the end, it was published, and one of the Russian researchers in the state bioweapons systems said, well, this is not a surprise. So either he meant he expected it to happen, or he already knew. I mean, this is quite possible that there are in Fort Detrick or in one of the Russian centers, there are forms of smallpox that could evade vaccination. So these are, I mean, these are very worrying things. The government in the US started pouring money in to gain-of-function research. This is all funded for biosecurity and military security reasons. How bad could it be if the bad guys decided to do this? Then, as the people realized that, well, if terrorists are going to cause havoc, they don't necessarily need an airplane or a modified bacterium. They can do it with a truck or with knives or whatever. The political military security aspect receded, and the focus became on, well, partly because there were a series of mini pandemics, which they weren't pandemics, mini outbreaks of viruses transmitting into humans at the beginning. There were MERS and SARS, all of which were MERS in particular is worse than COVID, but has been able to be, and it's still out there, but it's been able to be contained by basic public health measures. Similarly, SARS was able to be stopped. But these were warnings, and the epidemiologists and the virologists said, look, you need to enable us, this funding you've been given for kind of crazy biosecurity or political military reasons, forget about that. We can help prevent or understand better the possibilities of the next pandemic. And I mean, this is not bad, and not a bad idea in and of itself.
And Matthew, you said that the Russians weren't surprised. What have we learned from Russian defectors? I mean, I've read some of Ken Alibek and
well, Ken Alibek was the big. So from 1975, I mean, there were five Russians at Asilomar, and they were accompanied by what was described as a very dapper attaché from the San Francisco consulate. Now, the San Francisco consulate was shut down in the 90s as a nest of spies, which indeed it was. And you don't need to be John le Carré, the thriller writer, to know why this particular dapper attaché was hanging around with the Russian scientists. So he was clearly some kind of minder stroke spy. And the Americans' impression of these old men, because they were old, some of them were in their 80s, high up academicians in the Soviet Academy of Sciences, their impression was that they knew nothing, that they had no idea of what was happening. And they were kind of laughed at as kind of bumbling idiots. There's one story about so there's a slide being projected, and one of them gets up and he gets his camera, which has got a little flash cube, because we had flash cubes in those days, and he takes a flash picture of it. And of course, all he's going to see is a blank screen because the flash is going to wash out all the images. So this was a joke of how bumbling and stupid they were. Well, I think John le Carré might have called that good cover, because we now know that three of the people who were at Asilomar, these leading scientists, were also involved in the Soviet bioweapons program, which had already begun, had already been funded. And basically, they made a terrible pact with Brezhnev, the Soviet premier. They said, because Soviet genetics had been destroyed by the Lysenko affair in the 1940s and 50s, when it was all politicized and genetics wasn't real and we couldn't have any genetics. So basically they said to the government, okay, you give us a lot of money to develop molecular biology and we'll give you some new weapons, which is what they did. Now, the West had no idea about any of this. They had various crazy ideas. So in the 1980s, there was all sorts of stuff about yellow rain. The CIA became convinced that in Southeast Asia, the Soviets were spraying these genetically modified somethings because of all these yellow spots on the leaves. And they got very agitated about that. There are huge articles in the Wall Street Journal. And, obviously these articles were being placed by the CIA to kind of build up fever in Washington about the threat from the Soviets. Turned out all this stuff was bee crap. So after the bees have eaten collect a load of pollen and stuff, they tend to crab out yellow crap. But at the end of the 1980s, when a series of Soviet scientists from this program defect to the West, then the West is absolutely terrified, because they had no idea that this whole program had been going on. So Ken Alibek was the most public-facing one, because after 5 years of debriefing, took a long time, he was finally allowed to surface, and there was a big article in the New Yorker, and then he wrote an absolutely terrifying book called Biohazard, which is, I haven't got any hair left, but it makes the hair stand on, and because he describes all the various things that they got up to. And he was one of the leaders of this project. And he's still an active scientist in the US, but he's not engaged in anything shady like that anymore.
Mm-hmm. So we've talked about some of these, physical dangers and things like that. It also seems, to get back to ethics, money like that incentives and managing the incentives, patent law around all of this has been a big thing. What did you say was Jim Watson's biggest contribution, he believes?
Well, I asked him because he was starting to say some various racist things. So I tried to get him change the subject. So I said, Jim, what's the most important thing that you ever did? And he said, this was about 10 years ago. He said, well, it wasn't discovering the double helix, because that was inevitable. That would have happened, whatever. If we hadn't found it, then Franklin would have done, or Wilkins, or Linus Pauling, or somebody else was going to find it, which is actually true, right? I mean, it's, they got there, they stumbled over it, as Crick put it. So I said, okay, so what was it? And he said it was opposing the patenting of human genes. So at the early stages of the Human Genome Project in the 1990s, there was a big argument about whether human genes could be patented. Watson was absolutely opposed to the idea, and at the time, US patent law allowed this, but he thought it was an immoral thing to do. And I said, why did you say this? Why did you take such a stance? Because in the end, it ended him, and he was running the Human Genome Project, and clearly, in a way, this was kind of the culmination of his whole life's work. And then he's effectively thrown out. He has to resign. His position becomes untenable. So it wasn't just a position written on paper. It had real consequences for his life. I said, why did you take that stance? And he said to me, I'm a socialist. I said, oh, right, that's not new. Well, can you explain what you mean? And he said, well, I believe in universal healthcare and universal education, which I guess in the US makes you a bit of a lefty. I'm not sure whether he still holds to those views, but that's what he said to me 10 years ago. So the issue of patenting is absolutely fundamental to the explosion of the biotech industry, because up until 1980, you could not patent any living thing, with the exception, because patent law always has exceptions where there's money to be made. I mean, it's not actually, it's just made up by people, by lawyers and judges. It's not rooted in anything kind of coherent. It's just, does it make sense in the law? So US patent law forbade the patenting of anything living, with the exception of various hybrid plants, which is how the US seed industry, not insignificant for US agriculture, became so powerful and able to produce hybrids between two different kinds of plant, and then you could get huge productivity gains. This was one of the things that happened after the Second World War, well, from the 1930s onwards, but in particular after the Second World War, that led to the explosion. Our ability to feed all the people on the planet, So that was allowed, but you couldn't patent a gene, you couldn't patent a microbe. And in 1980, there was a court case finally reached the Supreme Court, and it had been rumbling on, these cases, except when suddenly the Supreme Court decides it's urgent. These court cases generally take a very, very long time. And all the investment and the hopes pinned on genetic engineering had been kind of in abeyance whilst this case finally came to the Supreme Court. And the Supreme Court decided, and this was in fact about a test case about a researcher who developed a microbe that could digest crude oil, so you could chuck it in the ocean and it would eat up an oil spill. It still hasn't been applied, but, that was the idea. And he wanted to patent this. He worked for General Electric. And so this court case eventually was approved by the Supreme Court. They said, yes, you can patent your little microbe. And the implication being you can patent anything that's alive. So that held in the US from 1980 until about 2013, when the Supreme Court changed its mind again and said, no, you can't do this. But it led to patents being taken out, change in the scientific world, because all of a sudden, researchers wouldn't talk to each other about what they were doing because they had a financial investment in it. They had signed an NDA. They couldn't discuss it with anybody. Their little startup that their boss had created would not tell people what they were doing. When researchers went to conferences, they wouldn't present their latest results because a competitor might be able to get to it. So this led to a lot of money being made, in particular by the people who came up with the basic technique of cloning, which is still used today, and made a lot of money for them and a lot of money for universities. And it was then licensed out to people like Genentech, who made even more money.
So Matthew, we're talking about incentives, law, money. A lot of this hangs on governments. What should we be thinking about when it comes to China and genetic engineering? I mean, we know that the culture there, a lot of secrecy, the Jiankui issue.
Well, yeah, I mean, there clearly is, China has had a very strong eugenicist tradition. That is, they think we should do what we can to improve people. But then, Many countries have, including the USA, which, implemented eugenicist laws in the 1920s. Indeed, they were the model for the Nazi laws. So this is something that is, present in countries all over the world. But it's very clearly the case that He Jiankui thought that he had the approval of the Chinese state to go ahead, and he certainly had the approval of some local officials. He was, had been headhunted and was building this was key in this new institute that's being created. And initially, the China Daily hailed this result as being a great victory for Chinese science and how marvelous it was. And then within 12 hours, because of the outrage that was on Weibo, the Chinese equivalent of Twitter as it then was, from scientists and from the general public, they quickly backtracked and they said, okay, right, all that has been removed from the internet in the best traditions. So I think China is much more wary, certainly of human genetic engineering than it was. It has been very keen on plant genetic engineering. Indeed, the first genetically engineered plant was created in China. Crop was harvested in China, and it was tobacco. And the US would not allow it to be imported, not because they thought it might be carcinogenic, but because they were worried that it had been genetically engineered. This is in the late 1980s and early 1990s. And this was kind of done by, again, a bit like He Jiankui. It was done at a local level without any kind of national guidance. And one of the things that in researching for my book As Gods, I was particularly surprised to discover is that, you get the impression of China being this very monolithic situation in which people can't speak out, they can't act independently, or at least not without local support. But there's a fantastic book called GMO China by a Chinese researcher called Kong Kao, which describes the huge debates that have been taking place inside China over GM crops, in particular GM rice, the possibility of introducing GM rice. For very obvious reasons. This is an extremely significant issue in China. They eat a lot of rice. It's part of, an essential part of culture. And there's something strange goes on because it's the same a bit in Europe. Europe doesn't have a single staple food like that, but people are very concerned in the idea of eating something that, they have tampered with, whoever they are and whatever that tampering might involve. Perhaps it's improved. That runs very deep. And yet the idea of injecting yourself with something that has been constructed in a bacterium, which is what we do with insulin, that doesn't seem to be quite so problematic. And I'm not enough of a psychotherapist to know where that deep root, but it's deep in people's minds. And in China, this has led to astonishing debates, quite open debates, but within the leading figures of the Chinese party as to whether this is a good idea or whether it's a Western trap and it's all going to, the kind of crazier ideas about what it might do to our manhood and our vital essences that you hear over here are being repeated in China as well. And they even gave Greenpeace quite remarkable freedom to campaign against genetic engineering of plants. So, I think China is much more fluid than we might think, but its vastness, both geographical and obviously numerical, and therefore the power it has. It is the scientific powerhouse of the world now. And even more so because of what's happening in US science at the moment with the funding cuts, it will become so even more. I was in China recently, and they have a saying that they're very good from going 1 to 100. What they're not good at the moment is going from 0 to 1. So it's making those breakthroughs. That is their science has yet to kind of really master. And I guess it's partly the funding issue. And if you're not sure that it's going to work, then don't do it, right? Whereas risk-taking is more encouraged in the West. And indeed, for the startup culture, that's the whole basis of it, that maybe it won't work, but, maybe it will. So I think I'm not terribly worried about China. In a way, I'm more worried about the USA, right? So the USA, because there is no federal restriction on genetic engineering of human embryos. I mean, it's completely forbidden in Europe, in Australia, in many, and now in China. But in the USA, whilst there may be state restrictions, there's no federal restriction. The only federal restriction is on using stem cells with federal funding. So you can't do that. Whatever you're going to do with them, even if it's, completely academic, you can't use the stem cells. But if you are part of a private university with private funding, then, and your state allows it, you can do what you want. And indeed, there have been various kind of hints that there have been at least thoughts in this direction by various researchers in the USA.
Mm-hmm. And for those who don't know the differences in this sort of thing too, this plays out with in vitro fertilization laws. Mm-hmm. I mean, in the US, this is very common.
Yeah.
This is just a sort of widely accepted thing. How are the differences playing out around the world?
Well, yeah. So in the US, I mean, basically, well, there's in vitro fertilization, which is, as I said, generally described very glibly by male enthusiasts. But I would invite, any viewers or listeners there's a couple who've been through that, or who have a sister or a mother or an aunt who's been through, a friend will know that it is very, very tough. So it involves, harvesting, as they put it, lots and lots of eggs.
I mean, let's lay that out. I mean, what is that? There's hormones.
Yeah, you basically have
eggs.
to. Yeah, you have to be. You're turned into It's an agricultural procedure in a way, because what they want to do is to get the eggs out to then do various things with them. In the simplest procedure, simply fertilize them with your husband's sperm or your partner's sperm or a man's sperm. But getting eggs, I mean, normally you produce one a month, right? So you need many, many. And this involves rounds of hormonal procedures, which doesn't generally leave you feeling too good. It's not anodyne, and people who do it for very committed reasons. They desperately want a child. It's the most usual use of that. And I mean, this was invented in the UK, just down the road from here. The first test tube baby was born in a place called Oldham, north of Manchester, where I'm speaking from. And that changed the possibilities of being able to deal with infertility, really. So that was the initial aim. That was the aim of the researchers who developed this, the surgeons, the three people who eventually won the Nobel Prize for this. They wanted to deal with infertility, and by harvesting eggs and then by introducing sperm, even very kind of weak immotile sperm, you could still finally persuade them to penetrate the egg and to produce an embryo, which you then had to reimplant and hope that it would all go well. So that is widely used and is commercially available, for example, in the UK. But the next step is what's called pre-implantation selection. And this is where the ethics gets problematic. So in the normal run of things, you've got loads and loads of embryos, which you've fertilized, if you're lucky. You can implant some of them. In the old days, you tend to implant a lot of them because it might fail. And that's why you used to get lots of multiple births. They've they've improved the technique now, so you generally don't get, quintuplets or whatever born through IVF. But you do end up with lots of eggs left over. And so one of the things that people do is to then store them. Or women may choose, if they've got very busy careers, to do this at a particular moment of their life that suits them, go through IVF, have the embryos frozen, and then at a later stage they can implant them. Now, the issue comes, well, what I've just described is a bit like how you and I were made. It's just random stuff. You don't know which sperm is going to fertilize which egg and all the rest of it. And you get what you're given. You get a baby, it's a boy or a girl, and it's got a particular personality and looks and all the rest of it. And that's part of the magic of human reproduction. But what if you had a genetic disease in your family? And in particular, a really horrible disease like Huntington's disease, which is what's called a dominant one, so that if you have the disease, then your child will definitely have it. You will definitely transmit it if you can definitely transmit it. Then that is, you can detect the embryos that may be affected. And so one of the amazing things about human embryos is that they start off as a single cell and then they divide. And when they're at a certain stage, 8, 16, you can take one of those cells out. And it's not like each cell is destined to become an arm or a head or whatever. You can, the embryo will sort itself out. Interestingly, this works in certain class of animals. It doesn't work in all animals. So if I did this spent most of my career studying flies. If I did this in one of my flies, the fly would die. Fly embryo wouldn't work. But in a human embryo, you can take one out and one of these cells out and therefore sequence it and see, well, what is the genetic characteristic of this particular individual? Now, you could start off, as we do in the UK, by saying that this pre-implantation screening is acceptable for certain very, very unpleasant genetic diseases. And what you would do is you would then screen through the embryos. Okay, those have all got the defect. We don't want them. Those are okay. We're going to implant them. So in the UK, that is very, very restricted, but it is allowable under very strict control. On the other hand, you might say, well, okay, I really want a boy or I really want a girl. You could do the same thing. That is illegal, but I believe it's allowed in the US. You can just go through them. 50% of them are boys, 50% of them are girls. Yeah, get rid of them. You could then say, and this is what the big excitement is, that you could actually, in the random nonsense that there is about making babies, some of the embryos are destined perhaps to be a bit taller or a bit shorter or a bit, who knows, a bit cleverer. And the claim is that you can link genes to destiny. And unfortunately, this is not true. And you would have to increase the probability that your baby was going to be about an inch higher. I think the calculations, you have to have about 10,000 eggs. So many of the things that people are excited about, having a taller baby, feed them better food. The world's tallest nation are the Dutch at the moment. And that's not because they've had their genes altered. It's because they've been eating lots of food. You can see that, if you're in Europe, go to a museum and you'll see knights armor for knights. And they're all for guys who are about 5 foot nothing. And that's not because they were all kids who were fighting. It's because they ate crap food and so they weren't very tall. Yeah. So you can see that there's a kind of, it's not a slippery slope, but there are gradations to this pre-implantation screening. And that's where the money is at the moment, because there are lots of claims. My advice, don't waste your money. Have the baby in the normal way if you can. That you can improve your child's destiny by selecting certain combinations of genes. I mean, this is not true, but people want to believe it, so it's going ahead. But again, in the UK and in Europe, this would be completely illegal. You would not be able to do this.
So we're still a ways from the movie Gattaca, but we've got these gradations around the world in the way that different countries are handling these things.
Yeah, I mean, I think we're away from Gattaca because I think that's science fiction, i.e. the underlying idea of having a destiny. I mean, genes are destiny in some cases. With Huntington's disease, it is destiny. But even then, fantastic news about 2 weeks ago that they actually made the first step to an effective treatment in a patient of this horrible disease that is basically your brain is kind of falls apart when you're late 30s, early 40s. Very, very nasty neurodegenerative disease. But even then, it may not be destiny with modern medicine and with the use of understanding what the gene does and how it alters the growth and death of neurons in the brain. So the same thing with sickle cell disease. Sickle cell disease is, if you have 2 copies of the sickle cell gene, is very, very debilitating, extremely painful, very nasty. But there are now treatments, both pharmaceutical and just beginning, very expensive genetic engineering techniques, which will alter the blood cells in your body and mean that you no longer have this disease, or you no longer have it to a detectable extent. So even genetic diseases aren't, ineluctable destiny with science. The things that people are interested in, like intelligence and height and, I don't know what, sense of humor or athletic ability, I mean, these are either largely environmental, overwhelmingly environmental, for example, in the case of height. And the genes that may have an influence or cause a predisposition there are lots and lots of them, each having a tiny, tiny effect. So the idea of being able to, select, okay, I want a basketball player for a child, then, I say you're much better off just feeding them well rather than hoping you're going to find some set of combinations of A's and C's and T's and G's that will give you a tall child.
So we have this line between selecting from many to try and optimize for these factors, and then using CRISPR, theoretically these genetic scissors, to get in and make changes. And that's the Jiankui issue and things. So what's that distinction there?
Well, so what we would have done, or what you can do in America, is to select your embryo with the promise that it will have certain characteristics, or is more likely to have certain characteristics. The idea behind genetic engineering of humans will be just the same as genetic engineering of a plant. It would be to introduce a change that would make a definitive alteration to the physiology of the offspring and would be passed down the generations, because you are altering every cell in the body. You're not just, say, altering the blood cells, which is what you would be doing if you were going to cure sickle cell disease. You would alter every cell in the body, and above all the sex cells, the eggs and the sperm, so it would be transmitted to the next generation. So that's the fundamental difference, is that what you're doing is for life and for the future, right? It's not just for one individual. You are changing something forever. Now,
it's going to get passed on.
Yeah, we've done this for generations for millennia with plants and animals, right? That's how evolution works. Never mind agriculture. When we're out chasing animals, then the animals that are slow, for one tiny proportion of which will be due to their genes, we're going to eat, and so we're going to end up with faster and faster prey. We've got to be clever and clever. I mean, that's whether it's a human doing this or a jaguar, it's all the same. So animals and organisms interact and alter each other's genes. But the difference here is we'd be doing it in a directed way. We think what we were doing. Now, what Jiankui said he wanted to do was to introduce mutations that would prevent these babies from getting HIV. And there's lots of reasons why he chose HIV, primarily because in China, people with HIV are heavily stigmatized. And although if you go through IVF, if you have HIV, then it's okay. It's not transmitted because, put it very simply, basically, the eggs and the sperm are cleaned, but in the procedure, and so there's no virus associated with it, and so you can have a healthy baby. The embryo no longer has HIV in it. If it's going to be reimplanted into the mother, and the mother has HIV, then that's a different story. But what he wanted to do was to introduce mutations which are known to be associated with a reduction in the infectivity or in your susceptibility to HIV. Unfortunately, these mutations, and they're observed in people, because we know there have always been cases, of people who've had, had loads and loads of unprotected sex or used IVF, used IV drugs, and never got HIV. And some of these can be identified, reasons can be linked to a certain number of mutations. But those mutations also have negative effects. You're much more likely to be susceptible to various other diseases. So it's not a win-win. You don't just change everything. And the problem came so as you said, CRISPR is these genetic scissors which were invented in 2012 and shown almost immediately to be applicable not only to microbes but also to human cells. So you can do astonishing things with them. But it turns out that whilst you can do these experiments and alter at will sequences of DNA, so for example, changing the Huntington's disease gene so that it no longer produces this terrible effect, or altering a single base, the single letter of DNA that causes sickle cell disease, although that's technically possible in an embryo, when you actually do that in an embryo, it turns out that the CRISPR techniques we're using at the moment have really very, very alarming side effects, that you can lose whole chromosomes, that rather than being a pair of scissors like you describe it, it's much more like a kind of chainsaw run amok. And this is because of the specificities of primate embryos. So most of these experiments have been done in primates and then occasionally in embryos that have been overproduced in IVF. So they're human embryos which aren't going to be used. And when they're at the initial stages, even in the UK, they can still be studied. And you can see what happens. And what happens is that it's not just changing one base pair or removing a small amount, then whole bits of DNA get lost during the process. And this is what happened with Jiankui. He introduced these mutations, he thought. Not every cell in the body is affected in these girls. We know that from the sequence data that he presented. So they're mosaic. So some of their cells have got mutation, others don't, which is not normal. This should not be happening. The reason why that happened is that when he injects all the CRISPR gubbins into the embryo, which is a single cell after fertilization, the cell's not sitting there. The embryo's not sitting there. It's going places. It wants to become something. You know, there are bits of it moving around all over. And so whilst you've got this CRISPR chemicals, you know, enzymes and so on, trying to find bits of DNA and chop them up, the cell itself is doing something else. So he's ended up with mosaic children. And above all, the mutations, he didn't actually introduce the mutations he was hoping for. And at least one of the mutations he's introduced has never been seen in any human. I mean, we've got millions and millions of genome sequenced, and this particular mutation has never been seen in any human. Now, with luck, that will be okay for them. None of this, but it may well be that something horrible has gone wrong. So that's why, never mind about the ethics of it. This is, it's incredibly unsafe. And I don't think anybody but the most kind of heartless, tech evangelist would think that this was a sensible way to go.
Right. And then what did David Liu from Harvard say?
When Jiankui announced this at this big jamboree in 2017, David Liu, who's an amazingly smart guy and has developed a much more delicate single base pair change technique called prime editing, which doesn't seem to have all these horrendous side effects. Anyway, he just asked the killer question. He said to Jiankui, what was the unmet medical need? What were you curing in these girls? Because you've got to remember, these girls were perfectly normal. They were just like any Chinese, or the embryos, they were just any Chinese embryos until he got his hands on them and started messing around. They had no medical need. This was he claimed, a prophylactic procedure. Well, we all know how not to get HIV. It's well known. They teach it in high school, and you don't need to edit your genes to do it. Right. So yeah, David Liu's comment, that just shows that this particular approach, this particular claim was absurd. Now, I could imagine that he might have done it with, say, sickle cell disease, with a patient with an embryo that had sickle cell disease, or with somebody who had Huntington's disease. So it's not necessarily absurd, but I think that it is, the number of people who could be saved by, bioethicists have worked out that there are various genetic diseases that you might want to cure that are relatively frequent, including Huntington's disease. But the number of people whose lives you would enable, right? Because you're not saving any lives, because there are no lives until you put the egg and sperm together in the test tube, yeah? But the number of healthy lives or unaffected lives you might enable would be numbering in the few hundreds, that's all. And really what you're treating in this case, if you're not doing it for aesthetic or rather crazed reasons about eugenic reasons for what you want super race to be created. The real thing you're treating as a physician, if you were involved in this, is not actually the future embryo. It's the parents, it's the patients. They want to be parents. They want to have a biological child of their own that is not affected by this genetic disease. And whilst, one's heart goes out to them for that, there's no right to have a baby or no right to have a healthy baby. And, there are lots of babies around the world and children who don't have parents who need adopting, and they may be healthy and they may be not. And my own advice to those parents, which is kind of useless, but would be that's maybe where you should spend your love and your money, rather than imagining that we should be changing, taking this very dangerous, what I think is ethically and technically a very dangerous step by allowing such gene editing of genetic diseases. So even in the strongest ethical case, right, which is genetic diseases, I think there's now an acceptance that this is just, there aren't very many people who would benefit from this, the parents, right? And we know where they are. We know who they are, right? This is not people in an African village or in South America, favela, who are going to benefit from this. This is going to be for the incredibly wealthy, the people who can afford it, because it is amazingly expensive. And we know which countries, and we can probably have a fairly good guess about what color they're going to be. So this is in fact changing our views of the world for a for the benefit of a very, very small minority.
Yeah. How do you think that advancements in this technology will affect inequality in the way that it moves?
Well, I probably not. I'm not actually that worried about it. Firstly, because it takes a long time. So the idea of Gattaca or of Brave New World, or of the Master Race, right? Look, it takes it takes what, 25 years, generation time. Views of what is, beautiful or what is desirable can change in that time. And it would take so long to actually ship, plus people like having babies the old way, In general, it works quite well. And there's a bit of magic associated with it. And when it doesn't work, well, we can try and fix that. But, I'm not terribly worried about the creation of a master race. If it were to happen, it's going to be centuries away. I'll be long dead. I don't think that's terribly worrying. I think it's more what's concerning is, there may be, I guess you've also got to think of the children, right? The children get thought of, and we've had this already with children who are born in order to donate an organ to another child who's affected. So effectively they are being chosen as a vehicle to donate either blood or, a kidney or something to some child. And this is, I don't know how that would make me feel. Maybe you're the savior. Maybe you're just, a bit of meat that happens to have the right DNA. I think that, nobody has to be born, but to be born as part of your parents' desire for, parents put an awful lot of pressure on children anyway. You're going to be, you can imagine the rows, the family rows when they're teenagers. I hate you. I didn't ask to be born. Yeah, well, I wanted a baseball player or whatever. I wanted the next Einstein or Mozart, and you can't even play a piano. So this could actually be really destructive of those family. And I'm not being entirely facetious. You just got to think about what it would mean. I don't think this is going to have a massive effect on society, except in the sense that it encourages the idea that DNA is destiny and that we are in some way all beholden and programmed by our genes and that there's nothing you can do to escape it. Whereas in the vast majority of cases, that is just not true. And you are made by a magical combination of your environment and lots of bazillions of tiny little influences of DNA, which will change over time and in the environment that you're in.
Yeah. Matthew Cobb, this field and your work is so interesting. Before we get to our last topic, for people who want to know more, where should they look for you online?
Well, they can find me on Blue Sky. I'm Matthew Cobb. I have a dormant account on Twitter or X, so I wouldn't go there. You can find my books at I would go to bookshop.org or uk.bookshop.org if you're in the UK. And you can find various books. In particular, the most recent one dealing with this is As Gods. It's the fancy American title. There you go. The UK title is rather more sober, The Genetic Age, but it is the inside of the book is identical.
Great. We'll put some links in the description to those and to your newest book.
Yeah, my newest book, you can see it over here. This is Francis Crick. This is the UK cover. This is a big biography of Francis Crick, who actually had virtually nothing to say about this. When genetic engineering was first being discussed in 1971, he was at a meeting and they had a big beery session in the evening all about it. And Paul Berg, who's the guy who'd come up with the idea, the young people were very concerned about, the young researchers at this summer school. It was on in an Italian castle in the evening. They're up on the turrets and they're having this big argument. And Crick just isn't paying attention. It wasn't his thing. He'd moved on. He was thinking about a very crazy idea he had about chromosomes, and he just didn't pay any attention to it.
Yeah. Well,
but there's lots of other stuff in there.
Yeah, that leads into my last topic, really, which is, you have so much insight into the man of Francis Crick. And I think a lot of us know him through that first line of the double helix, which is, written by Jim Watson, which is, I have never seen Francis Crick in a modest mood.
Yeah.
What do you think of that line?
Well, it's a great opening. Interestingly enough, initially it just said Francis. So in the first draft, the double helix, if people have not read it, they should read it. But do not believe every word you are reading. It's a novelized account of events by Watson that he wrote in 1968. Crick was absolutely furious. He tried to take out a court case against it to stop it being published, him and Maurice Wilkins. This goes back to the generational thing I was mentioning, talking about earlier on, because whilst the young genetic engineers were younger than Watson, Watson was kind of intermediary between Crick and Wilkins, who were these pioneers of molecular genetics, who were all born during the First World War. So, they're a completely different generation to Watson, who's kind of 13, 14 years younger. And Watson wrote this very racy book, which is very exciting. And he wrote it to inspire people. He wrote it very deliberately as a page turn. He wanted to, so there's, he was inspired by, kind of semi-fictional accounts of true crime and so on, to write something that was going to grab young people in particular. In fact, he wrote two books. He wrote a textbook as well in 1965. At the same time he's writing the double helix, he writes this textbook, The Molecular Biology of the Gene. So that got the academic kids involved. The double helix was supposed to get everybody excited about science and so on. And Crick was furious because of this very intimate description, which that opening line, as I say, initially it just didn't have Crick, it was just Francis, at a time when scientists were, or at least as far as Crick was concerned, should be, addressed as Dr. Crick. He wasn't a professor then. Here we've got this, it's all Francis and Morris and Jim and Max and Rosalind. It's all first names. And this intimacy, which Watson brings over, was something that Crick, as quite a private person, who he'd like to keep his private life quiet, was quite upset about. So he and Wilkins, as I say, tried to take out a court case in the US to stop it being published, but it was no hoper. Partly because they showed it to a lawyer who, a lawyer was given it, and he gave it to a friend who knew Crick, and he said, that's him to a T. That's exactly what he's like. So, it is, in fact, I think it's a very accurate. It wasn't that he was not modest. It was just that he never stopped talking, which drove people crazy. But it meant that when you were interacting with him, you were going to have a good time. You were going to have ideas bounced out, and he would always have something to say. And that's what Watson loved with him. They didn't work together really for more than about a month or so. At the beginning of 1953, they were friends for much longer, but their intense collaboration. But this is what I described in the book, is Crick's habit, his need to have intense collaboration with other researchers, which he did throughout his life. For 20 years in particular, he worked with Sidney Brenner, a South African researcher, and they worked together in Cambridge. They shared an office, and they had what Brenner called mad sessions, in which for a couple of hours every morning, they would just yak at each other about absolutely everything. And we can actually see this kind of crystallized in the blackboard that they used. So they were leaving one particular building and it was being cleared out, and one of these blackboards was going to be put in the dumpster, and somebody went off and took a picture of it. In fact, in the UK edition, I'll just show you the end papers, as it's called, so the inside. So we can see this is Crick and Brenner, their mad session, and this is something to do with some DNA experiment, you can see the DNA letters here. It's all to do with some virus that they're studying, bacteriophage virus. So I think it's probably a fair introduction, but Watson's book is remarkable, but it's not all true. That's the main thing.
Well, Matthew Cobb, for those insights into this field, for your enthusiasm for it, for sharing it through your writing, and for taking this interview, thank you so much.
Thank you very much, Brad. It's been a great experience.